Okay, today we are going to talk a little bit about the reproductive systems of humans, and we're going to start our conversation primarily with the male reproductive system. But before we do that, we need to consider a few things. So we're talking about reproductive systems, and there are a few considerations regarding these systems that apply to both the male and the female. We need to remember that the purpose of these systems is to produce gametes. And the fancy term for the production of gametes is known as gametogenesis. For males, that is also more specifically known as spermatogenesis. And for females, oogenesis. Production of spermatozoa, production of an egg. And we'll talk about what's involved with those two processes, some similarities and some differences as we move along. Arguably, the purpose of our living organisms, of us as living organisms, is to produce more of ourselves, more of the same. And in order to do that, we need a reproductive system that can produce gametes. The other thing to consider is there has to be a delivery system for these gametes from the male system into the female system, and there has to be delivery within the female system to a location where the spermatocytes, spermatozoa can make contact with the egg and fertilize. So we've got to look at the pathways for these gametes in the two systems as well. Males produce sperm from puberty until death by the millions on a daily basis. Females, on the other hand, produce one egg per month from the onset of menstruation, usually somewhere around puberty, until menopause, somewhere 30 to 40 years after menstruation begins, it will stop. And during that reproductive lifetime for a female, about 400 viable eggs will be produced. The other consideration with respect to both reproductive systems are the gonads. In the male, that's the testes, plural, and in the female, ovaries. So we'll talk a little bit about those locations for these processes. And then we need to consider duct systems. That's our pathway. And any accessory glands that may or may not be important, well, in our case, are important to the process, essentially, of spermatogenesis and oogenesis. Okay, so let's take a look at this male reproductive system. So again, we're thinking about the organs or the testes, the ducts, and the glands that would be associated with the male system. The location of spermatogenesis is the testes. The testes are going to produce sperm and secrete some hormones and introduce those gametes, spermatozoa, into the duct system, where they can mature and be transported for the purposes of fertilization. And the glands that are going to support this process are going to be producing what's called seminal fluid. And that seminal fluid is designed to improve motility, movement, and nutrition for the developing spermatozoa. So that fluid plus the spermatozoa is what's described as semen. So let's take a closer look at these testes. If we're just looking at one, it's a testis. Testes is plural. We've got a location for these testes is outside the body proper, located in what's called the scrotum. And that's a pouch with a split, a septum that separates the right and the left, and is surrounded by an epithelium and a smooth muscle tissue layer, and then an outer sort of connective tissue layer for a little bit of protection. The muscular layer is called the dartos muscle, and when it's relaxed, it creates a very large surface area, relatively large surface area for the scrotum, which is going to allow the dissipation of heat. So what we're going to find out a little bit later is that the process of spermatogenesis needs to be a little bit cooler, maybe 3 to 4 or 5 degrees cooler than body temperature. So the scrotum is located outside of the core of the body for this purpose. The dartos muscle can regulate temperature by relaxing to allow temperature loss or contracting to better retain temperature. And there's another muscle that we'll talk about a little bit later when we talk a little bit more about the duct system. So this is a smooth muscle layer. So it can regulate surface area. So when the muscle contracts, it decreases surface area. And when it relaxes, it increases surface area. If we increase surface area, there'll be more heat loss. If we decrease surface area, less heat loss. So within the scrotum, the testes are located, and the testes are divided into sections that are called lobules. And there are several hundred of these lobules, anywhere from 200 to 300 lobules. And within each of those lobules, there will be found a tubule, 2 to 3 tubules called the seminiferous tubules. So the seminiferous tubules, 2 to 3, located within lobules, and there may be 200 to 300 of those lobules, all within one testis. So what we would have is a series of seminiferous tubules, maybe 3 within a lobule. And if we looked at the testes that are oval in shape and maybe about 5 centimeters in diameter, they would be formed from these lobules that would kind of set up as wedges. And within each of those individual wedges, hundreds of them, remember, there would be 2 to 3 seminiferous tubules. So you can see we're getting lots of surface area, lots of potential for time for the development of spermatogenesis. So the seminiferous tubules contain the cells that are going to become the gametes, spermatozoa, and those are initially referred to as spermatogonia, undifferentiated spermatozoa at that point. And also a cell type within the seminiferous tubule that's known as the Sertoli cell. So we have spermatogonia. We have Sertoli cells, and a better name, because that's a person's name, Sertoli, a better name for these, because it describes a little bit more of what they do, is a sustentacular cell. So the sustentacular cells, I'll spell that because my writing is terrible, SUSTENTACULAR, sustentacular cells. These are support cells for the process of spermatogenesis. In some older textbooks, they used to be referred to as nurse cells because they look after the spermatogonia and guide and support the process of spermatogenesis. Now, we're going to find out a little bit later that these seminiferous tubules are going to collect and form a series of tubules that are going to enter a structure that's called the epididymis. And the epididymis is within the testes. So all the seminiferous tubules are going to funnel towards the epididymis, and the spermatozoa that have been formed within the seminiferous tubules are going to be sent there to mature. We'll talk a little bit more about that when we look at the process of spermatogenesis. So what we're going to look at next is the actual pathway. Let me just black out. There we are. So we're looking at a lateral view of the male reproductive system. And here we have the testes. And within the testes, we would have the seminiferous tubules separated by some connective tissue within the testes that form lobules. These tubules are going to collect and funnel into the epididymis. And the epididymis is a coiled tubule that creates surface area and length and represents the location where the spermatozoa are going to mature. And they may stay in there for as long as 2 weeks, somewhere between 10 and 15 days in order to mature. If ejaculation occurs after that time, the mature spermatozoa will be pushed into the tubular system and ultimately leave the male body. So the pathway would follow through from the epididymis into what's called the ductus deferens. Let me use a brighter color here, maybe even just the brighter green. Okay, and this represents the beginning of our duct system that is going to return to the core of the body. So this is known as the ductus deferens or the vas deferens. You can see that vas or ductus deferens up here. And it's going to pass from the right or the left, or both, testes, and over the symphysis pubis, so over the joining of the two sides of the pelvis, and enter into the pelvic cavity, travel towards the bladder. So this structure that we see right here is our bladder, and travel superiorly over the bladder towards the posterior side of the bladder, where it joins with the what's called the seminal gland or the seminal vesicles, a gland that produces seminal fluid, and the ducts that empty the contents from this seminal vesicle join with the vas deferens to form the ejaculatory duct. So here we have the two contributions to the ejaculatory duct. At this point, everything is paired. So the vas deferens, there's a right and a left one, there's a right and a left seminal vesicle, right and left seminal ducts join to form a right and left ejaculatory duct. The ejaculatory duct is going to pass into the prostate gland. So this gland below the bladder and below the seminal glands is known as the prostate. And most of us have heard of the prostate gland. Now, what's going to happen within the prostate gland is that the two ejaculatory ducts are going to join with the urethra. The urethra is a tube system that drains the bladder into the penis for urination if we're following the urinary system, but joins with the urethra within the prostate gland to connect the duct system of the male reproductive system as well. So the urethra from the point of the prostate gland has a dual function, a function for the urinary system and a function for the male reproductive system. This is very different than what we're going to see in the female system. But what this represents now is a direct duct system from the testes to the organ that's responsible for ejaculation, the penis. Okay, so this represents a continuous duct system that will ultimately deliver spermatozoa from inside the body to outside the body. The final gland is the bulbourethral gland. So we have a prostate gland, the seminal glands or vesicles, and the bulbourethral gland. And this is our bulbourethral gland. So the three glands produce the semen, the seminal fluid. And the seminal fluid contains nutrients like fructose and vitamin C, and for nutrition and motility, and some prostaglandins, again, to encourage motility of the spermatozoa. If the spermatozoa are going to be considered viable, they have to be very motile. They have to have a lot of stored energy because it requires a lot of energy to propel from the male system into the female system in order for fertilization to occur. So the responsibility of the seminal fluid is to improve motility and protection of the spermatozoa, as well as nutrition. So the seminal vesicles, the prostate gland, and the bulbourethral gland contribute. And a couple of features. The seminal fluid is slightly basic because the fluids associated with the female reproductive system tend to be slightly acidic, and that is a negative factor on spermatozoa. So the seminal fluid will be overall a little bit basic. The seminal fluid is the major contribution, prostate gland the next most, and the bulbourethral gland the least. The bulbourethral gland, you can see by its location, contributes to the last part of the seminal fluid, maybe about 10%, 7 to 10% of total seminal fluid. And it's mostly designed to lubricate and neutralize acids from urine in the male system to encourage the motility and the passage of spermatozoa through the last part of that duct system in the male. Okay, now let's take a look at another picture here. So in this diagram here, what we're looking at is the testes, a cross-section of the testes. We can see the seminiferous tubules within a section or a lobule. If we take it out, we're going to look at a cross-section. We'll look at that in a minute. Okay, so all of these hundreds to thousands of seminiferous tubules are going to collect into a system of ducts here that is called the rete testis. And that's really just a coalescence of all the ducts from the various seminiferous tubules funneling into and ultimately entering the epididymis. So here's our epididymis, and we can see the coiled and convoluted pathway that's going to slow down flow and allow time for the maturation of the spermatozoa so that they gain their motility and their strength for the trip they may have ahead of them. And as it leaves the epididymis, it enters the vas deferens, and the vas deferens is going to enter the pelvic cavity through what's called the spermatic cord. So the spermatic cord would be an extension, and not only would we see the vas deferens within this spermatic cord, but we would also see blood vessels that are delivering blood to the testes and veins that would be taking the deoxygenated blood away from the testes and back towards the heart. And in addition to that, we would see some nerves. And all surrounding the outside, or within this layer, is a second muscle I mentioned earlier called the cremasteric muscle, or you may see it as a cremaster muscle, either way. And that's the muscle that is capable of retracting the testes closer to the abdominal cavity or the pelvic cavity. So in cold weather, the cremaster muscle and the dartos muscle will both contract to bring the testes closer to the body and reduce surface area in order to keep the temperature regulated at just maybe 2 to 3 degrees Celsius cooler than the internal temperature of the body. And in warmer temperature, the cremaster muscle and the dartos muscle would be found within the scrotal tissue, will both relax and allow the testes to move a little further away from the core of the body in order to keep that temperature, again, somewhere between that 2 to 3 degrees Celsius cooler than body temperature. So in this picture here, what we've done, if you can see it, I hope you can see it okay, we've got this cross-section of our seminiferous tubule. So here's our seminiferous tubule. And within the seminiferous tubule, there will be some nurse cells or sustentacular cells. They're depicted intermittently through there. And the rest of the cells that you can see in that bluey gray color are the spermatogonia cells. The spermatogonia cells are the cells that are going to give rise to the first to spermatocytes and then to spermatozoa. Here's our mature-looking spermatozoa. So the Sertoli cell, or the sustentacular cell, or the nurse cell, is located there, the one I just outlined in the color red. So it provides a number of functions. It provides some nutrition. It provides a stable environment for the process of spermatogenesis, which is all very important. And it forms remember in the brain we talked about a blood-brain barrier? In this case, the Sertoli cell produces or performs what's called a blood-testes barrier so that the immune system doesn't start to attack spermatozoa. Because you'll see in just a few minutes that when we change the structure of a spermatogonia cell, it no longer is considered a self cell necessarily, or will be recognized that way by the immune system. So it needs some protection. And that's part of the role that this Sertoli cell also performs. Now, what we don't see in this picture, and what's another important cell to recognize, is that between the seminiferous tubules, there are some cells that are called interstitial cells because they're between interstitial. And they were first identified by a guy named Leydig. So they're sometimes just called Leydig cells, but interstitial cells of Leydig indicate, or if you call them interstitial cells, then you recognize that they're going to be outside of the seminiferous tubules, and more specifically between the seminiferous tubules. So if we had a seminiferous tubule here and another seminiferous tubule here, then the interstitial cells would be found between them. So the Sertoli cells are within the seminiferous tubule, and the interstitial cells are outside of the seminiferous tubules. And that becomes important because hormones like LH that we mentioned and didn't really talk about, the LH hormone being produced by the anterior pituitary gland, if you remember from AMP 100, it stimulates the interstitial cells to produce testosterone. And testosterone, in turn, will target the Sertoli cell and increase the actions of the Sertoli cell in support of spermatogenesis, and also stimulates spermatogonia to undergo spermatogenesis. So there would be receptors on the cells of the spermatogonia and receptors on the cells of the Sertoli cell as well for testosterone, receptors on the interstitial cells for luteinizing hormone, LH, and receptors on the Sertoli cell for FSH, the other gonadotropin. I'm going to show you another diagram in a couple minutes that will help clarify all of that. But FSH is going to stimulate Sertoli function. LH is going to stimulate interstitial cell function to produce testosterone, which works together with FSH, so that would be synergistic on our Sertoli cells, and in the interest of producing spermatogonia to become spermatozoa through the process that we call generally spermatogenesis. So lots of terminology in there, but the concepts make good sense. Receptors have to be on these cells for these hormones to exert their effects. So the process of spermatogenesis is going to involve these steps that we can see here. So we see mitosis, meiosis 1, meiosis 2. And there's going to be another step that we're going to draw in here. And that step is called Spermiogenesis. Just to confuse you, that's one of the steps within spermatogenesis. So that's another piece of terminology that you need to become aware of. And I think we have a better diagram to show you that. Let's see. This is just a diagram of the cross-section of the seminiferous tubule. So you can see here that the letter B, that represents our Sertoli, or, sorry, that is not our Sertoli. I better get that right off of there. That cell that we just started to talk about, that's our spermatogonia. Those are undifferentiated cells that will eventually become spermatozoa. So in this diagram, you can see that the spermatogonia are pushed up against the outside of the inside, but the outside of the diameter of the seminiferous tubule, up against the outer edges. And these cells undergo mitosis. So they're going to undergo mitosis And when they undergo mitosis, two of the daughter cells, so if we looked at these two of those daughter cells, actually it's better if we look at these two. So if we had this cell, start with this cell, two daughter cells, one daughter cell is going to be pushed away from the basement or the basal layer, and the other is going to remain to be able to undergo continued mitosis. This one that we just introduced here would be known as the primary spermatocyte. So one becomes a primary spermatocyte, and the other daughter cell remains up, available for further mitotic divisions. The primary spermatocyte is going to enter into meiosis 1, the first division of meiosis. You may recall from high school or previous courses that mitosis gives us two identical daughter cells. In this case, one is going to be left behind in order to be able to undergo subsequent mitotic divisions, and one of the daughter cells is going to be introduced into the spermatogenic process. So that's the one that's going to enter into meiosis 1. This is a big target for testosterone. So our primary spermatocyte is undergoing meiosis 1. In meiosis 1, the genetic material is halved. Again, we'll talk a little bit about this probably next week when we talk a little bit about heredity. But most of us are aware that we're made up of 23 pairs of chromosomes. 22 pairs are described sometimes as autosomal, and the 23rd pair is the sex chromosomes, which are going to determine male and female anatomy and physiology. So in meiosis 1, the genetic material is going to be divided in half. And that's going to give us a secondary spermatocyte, actually 2. And they have half the genetic material each, and they will each undergo meiosis 2, the second half of the meiotic divisions, which is going to give us 1, 2, another division, 3, 4. These cells are going to be called spermatids. And at this point, these are immature spermatozoa. So they have to undergo differentiation. And when they undergo differentiation, they're going to develop a head and a body and a tail. A head and a body and a tail. A head, body and tail. Now these are our spermatozoa. Those spermatozoa are the ones that are going to travel from the seminiferous tubule to the epididymis. There they will mature in the epididymis for, say, 10 to 14 days and become effectively mobile or motile. They gain motility, they gain maturity, and enough energy to be prepared for ejection. In the male system, it's called ejaculation. And in the interest of fertilization, entry into the female system with enough energy and enough motility to travel through the female system to find the egg for fertilization to occur. So that process that we just described down here, where the spermatids differentiate to spermatozoa, that's the process I mentioned earlier that's called Spermiogenesis. So when we look at Let's see here. Is it going to show up? For spermatogenesis to be complete, we have to go through mitosis, meiosis 1, meiosis 2, and spermiogenesis. So the process of spermatogenesis really involves 4 distinct steps. 2 and 3 arguably are one, meiosis, but meiosis has 2 steps within it. So we're going to refer to these as 4 steps required for the process of spermatogenesis. And we're going to find out a little bit later that there's one more step the spermatozoa have to go through before they're going to become completely viable. But that 5th step is, well, that next step is not truly part of spermatogenesis, but it is part of viability, and it has to occur in the female system. So we'll talk about it when we talk about fertilization. So here, what we've got are the steps. We started with spermatogonia, undifferentiated spermatozoa, that begins the process of mitosis at puberty under the influence of FSH and LH. Specifically, FSH initiates the process of spermatogenesis by activating the nurse cells and changing the environment within the seminiferous tubules so that this mitotic divisions begin. So remember earlier I said that the process of spermatogenesis begins at puberty for males and continues throughout their lifetime. Now, it's not going to continue at the same rate as they get into their 7th, 8th, and 9th decades of life, but while they are particularly reproductive, so beginning at puberty and through 30s, 40s, and 50s, they will be producing spermatozoa at the rate of hundreds of millions per day, probably as many as 500 million spermatozoa on a daily basis. But we can appreciate that they have to go through these steps and stages. It's going to take a week or two to go through spermatogenesis and then be stored in the epididymis to gain more motility and maturity another 10 or 14 days. So probably in the vicinity of about a month or so, 3 to 4 weeks, 5 weeks, for the process of spermatogenesis to be completed and the spermatozoa to become ready for entry into the female system in the interest of fertilization. So the other thing to be aware of is that when spermatogonia undergo mitosis, they have the full complement of chromosomes. Still have 23 pairs when it's a primary spermatocyte. That was one of the daughter cells of mitosis. Then in meiosis 1, that genetic material is cut in half. So we're going to have 2 cells with half the mitotic, half the chromosome complement in each. So traditionally, in the jargon of heredity and genetics, the full complement would be described as a cell with 2N, where N is the number of chromosomes. In humans, it's 23. 2 sets of 23 chromosomes. And then the secondary oocytes, or the secondary spermatocytes, are 1N. So they underwent meiosis 1. And then they're each going to divide into, remember we call these spermatids. They will have half the chromosome material, but now there's 4 of them. And when they undergo spermiogenesis, we're going to end up with 4 motile spermatozoa, but not yet mature. They have to go to the epididymis and spend time there growing and gaining motility and maturity. And the other thing to mention, you notice here in this diagram, in each, I drew a little cap. That's literally called a capsid. And it's full of enzymes. These enzymes are necessary for fertilization. They have to be released to erode some of the barrier around the outside of the ovum in the interest of fertilization. So we'll talk more about that again when we talk about fertilization. But that's part of the maturation process during spermiogenesis. We gain a head with a cap. The body is this sort of coiled segment I drew here, full of mitochondria. Everybody remembers what mitochondria are for: the production of ATP, which it gives us our source of potential energy. When we break the ATP down to ADP, we get a phosphate group, we get some energy, we get some heat, but that's going to provide the energy that makes the tail motile. So it's a very, very unique, specific cell. This cell typically, or technically, would be considered to be foreign because it doesn't have the same genetic makeup as a self cell would, and that's part of the role of our nurse cell. So in our diagram here, you can see this nurse cell has a large amount of cytoplasm, and that cytoplasm provides nutrient and guidance for the direction of spermatogenesis towards the lumen. Once these are entered into the lumen, they are mobile enough to funnel into the epididymis. So remember our pathway for our epididymis. So here we have our Sertoli cell guiding the process of spermatogenesis from a spermatogonia to a primary to a secondary, spermatids, spermatozoa. Spermatozoa are going to be delivered to the epididymis. From the epididymis, after maturation, they are going to enter the ductus deferens, travel through the ductus deferens, and ultimately, if ejaculation occurs, the spermatozoa will be entered into the female reproductive system for delivery and fertilization possibilities. If no ejaculation occurs, the spermatozoa that were being matured in the epididymis are going to deteriorate. So they'll degrade after a few weeks of being stored in the epididymis in the interest of, in the event of celibacy or no ejaculation for fertilization purposes. So that's the end fate of spermatozoa. They either degrade are broken down by macrophages, and some of the nutrients can be recycled, otherwise removed, or ejaculation in the interest of fertilization. Those are your sort of fates for the spermatozoa that are being produced this way. Just another diagram of the same thing, a little bit larger, maybe a little bit easier to see. So we can see our Sertoli cell in this diagram. We see the steps of mitosis, meiosis 1, meiosis 2. This would be our Spermiogenesis. Okay, so my screen just froze up. I put us on a pause. Probably didn't really notice that. But here, down here would be our spermiogenesis step. And from there, we're going to travel into the epididymis after maturation and gaining motility, delivery through that pathway. So we would have our interstitial cells of Leydig outside, and they would provide the targets for our hormones. Let's take a look at this little diagram here. So here's where we can put it kind of all together with respect to the processes of spermatogenesis. Here we have our interstitial cells of Leydig. See, they're in the interstitium between the seminiferous tubules. There's the boundary of the seminiferous tubule. These are the cells undergoing mitosis. Sertoli cells surround, their cytoplasm surrounds the cells undergoing spermatogenesis. This is the nucleus of our nurse cell and the nucleolus. So there's a process of spermatogenesis going on here. Spermatids, spermatozoa, lumen of the seminiferous tubules, delivery to the epididymis. If we follow our hypothalamic pituitary axis, gonadotropin releasing hormone targets the anterior pituitary gland, and we've labeled the FSH as a purple square, and LH is identified by an orange triangle, so you can see specific receptors. These are not steroid-based hormones, so they need receptors on the cell membrane to activate the inside of that cell. So here we can see that FSH targets a receptor on our Sertoli cell. Purple receptor matches, activates inside, and activates the nurse cell to initiate the process of spermatogenesis. That LH and FSH both indirectly affect spermatogenesis. FSH under the influence, testosterone, Sertoli cells, androgen-binding proteins. So what we're going to get here is activation of the nurse cell, activation of spermatogenic cells, spermatozoa, by testosterone. Okay, round receptor on the nucleus of our Sertoli cell. We would see the same receptor cells on the nucleus of the spermatogenic or spermatogonia cells. So they begin mitosis. The nurse cell begins care and direction of spermatogenesis, and we end up with what we're looking for. If we follow LH receptors on the Leydig cells, there's our Leydig cells. Okay, match up with receptors on the outside of the cell, activate these cells to produce testosterone. Testosterone is released, targets Sertoli cells, targets our spermatozoa, and we initiate the production of ultimately our spermatozoa. Now, the other thing just to be aware of, this is an androgen binding protein, and it's responsible for binding and maintaining high levels of testosterone close to these spermatogenic cells so that the process of spermatogenesis is maintained. So FSH and testosterone together stimulate Sertoli cells to produce this ABP, and that promotes the production of spermatozoa. Hopefully that process and those steps are clear. You can certainly ask questions about all this stuff when we get together next class. Okay, so spermatogenesis occurs in the seminiferous tubules, enters the epididymis, and we said the epididymis was that coiled structure our whoops, a little bit of dyslexia there. Epididymis That's our site for sperm maturity. We acquire more motility and viability here. It takes somewhere between 10 and 14 days. It's a smooth muscle duct that stores the sperm and contracts when the male is aroused and initiates peristalsis. That's the term that's used that's going to drive the spermatozoa from the epididymis into the vas deferens. And as I mentioned, the sperm can remain viable in the epididymis for several months before being phagocytosed if no ejaculation occurs. So we get maturity. And viability occurring in the epididymis. Okay, and then entry from the epididymis to the vas deferens. Some of you might recognize the term vas from vasectomy. And a vasectomy is a method of a surgical procedure that results in male infertility, because you can appreciate that if we block the vas deferens, we block the delivery of spermatozoa into the rest of the duct system. So the fluids can still be produced and ejaculate will still occur, but without spermatozoa, because the vas deferens has been blocked in the case of that surgical procedure or any other blockage for any other reason. But most typically, it's going to be a surgical procedure, a choice that's made by the male. So the ductus deferens, of course, is much less coiled. So it's more of a delivery system at this point. It's an extension of the epididymis. It's continuous from the testes. So the whole duct system in the male system is continuous from seminiferous tubules into that rete testi that I mentioned that funnels into the epididymis, and that's tightly coiled and then uncoils as it becomes the vas deferens. Enters the pelvic cavity to loop over and then down the posterior surface of the urinary bladder to join up with the seminal vesicles, duct system from the seminal vesicles. So the seminal vesicles are going to contribute now seminal fluid, about 60% of the total seminal fluid. Remember, you don't need to remember percentages in my class, but recognize that if it's 60%, most of the seminal fluid is going to come from these seminal vesicles. So that's going to provide some nutrition for further motility and viability, as well as some protection. And this solution was primarily alkaline to neutralize some of the acids that were associated with exposure to urine and female reproductive system in the interest, again, of fertilization. So from the seminal vesicles, now we're going to become, as it leaves the seminal vesicles, it's identified now as an ejaculatory duct. There's still two of them, right and left side. And that's the duct that's going to enter the prostate gland and join with the ducts of the prostate gland to then join into the urethra that leaves the bladder. Once it connects to the urethra, we're at a single duct. The right and left ejaculatory ducts join to the urethra and become a single duct that's going to ultimately travel through the penis with dual responsibilities. In the case of urine removal, that's a function, or ejaculation, reproductive function. So the prostate gland, again, contributes about 30%. Remember earlier I said bulbourethral was give or take 10%. So nice, easy numbers, 60, 30, 10 in terms of contribution to seminal fluid. The ejaculate then becomes a combination of seminal fluid, So let's just complete this here. So the vas deferens plus the seminal vesicle ducts, vesicles, that's not there. Whoops. Let's get rid of that. That should just be the letter I in that space. Vesicles, that's what's going to give us the ejaculatory duct. If we add the prostate gland ducts, then we get the urethra. There's one urethra, there's two ejaculatory ducts, and there's two vas deferens. Two testes located in the scrotum, location for spermatogenesis within the seminiferous tubules, entry into the epididymis, vas deferens, right, left, two. Ejaculatory duct, right, left, into the prostate, receive content from the prostate, enter the urethra, then the duct becomes one. Seminal fluid, plus the spermatozoa is what the ejaculate is described. And a typical ejaculate will be anywhere from 2.5 to 5 milliliters of volume, and usually contains about 50 to 150 million spermatozoa per milliliter. If there's less than 20 million spermatozoa per milliliter, that's considered to be male infertility. So less than 20 million So that's going to give you an appreciation of the number of spermatozoa that are going to be required for fertilization to occur. If we don't really start with hundreds of millions of spermatozoa, the chances of fertilization, at least from the male side of things, is going to be pretty remote. So, and we'll reinforce that when we talk a little bit more about fertilization. So you can begin to appreciate the numbers that are required for this process. Okay, so we've talked about the testes. We know that the testes are oval in shape and about 5 centimeters in diameter and are divided into lobules. Within the lobules are the seminiferous tubules. Seminiferous tubules are the location for spermatogenesis. They drain into the epididymis. The epididymis is a structure that's on the outside of the testes, coiled, and leaves as the vas deferens through the spermatic cord. The duct system is ultimately delivering spermatozoa to the penis. In male arousal, the penis becomes erect. The erection occurs because of blood flow during the arousal phase into chambers that receive a relatively large volume of blood. They are called corpora, plural. And we have 3 of those. 2 of them are known as the corpora cavernosa. And one is known as the corpora, or singular would be corpus spongiosum. So this is significant because the corpus spongiosum is going to contain the urethra. So if this represents the urethra, the chamber, the corpus spongiosum, is located and surrounds the urethra. So the idea here is that during erection, the corpora cavernosa, they fill up to create stiffness, and the corpus spongiosum doesn't fill up quite so much, so it allows the urethra to stay open. That's pretty important if ejaculation is the ultimate goal. So this is under the influence of, let's put a variation of this color in. So outside of that, we would have the corpus cavernosa. The parasympathetic nervous system, PSNS, is dominant for erection, and the sympathetic nervous system is dominant for ejaculation. So there's one other thing that's important to recognize, or at least to note, with respect to the sympathetic nervous system. Remember, we said the duct system basically travels over and around. So here's the ductus deferens, travels behind, joins up with the seminal vesicles, becomes the ejaculatory duct, joins up with the prostate, and ultimately becomes one duct with the urethra. Now, the urethra has two functions. It empties the bladder for urination, or it becomes the pathway for ejaculation. So the sympathetic division is responsible for ejaculation, and at the same time makes sure that the valve that separates the bladder from the urethra stays closed during ejaculation. So sympathetic nervous system will be responsible for the propulsion of the ejaculate and close the valve between the bladder and the urethra. So that's important. That's important to remember so that the urine doesn't mix with the ejaculate. Okay, and the duct is able to stay open. The urethra is able to stay open because of this relationship between the corpus spongiosum and the corpora cavernosa. And this is an example where the parasympathetic nervous system and the sympathetic nervous system work together for the sexual response, rather than being antagonistic, as we've seen, for example, in heart rate, where parasympathetic slows and sympathetic increases heart rate, that type of thing. Okay, so I think we have enough information here to review the male system so that we can begin to compare when we look at the female system.